Good afternoon, everyone. Welcome to this session. And this is the initiative of BDG Lifesciences, which is called Envision with BDG. And I hope that I can see that many are the participants who have been regularly attending our sessions under this Envision with BDG initiative. And today is the sixth session, and this session is on brain investigation through eye, understanding the molecular mechanisms in neurodegenerative disorders. Now, this session will be taken by my mentor and the person who has taught me molecular docking 10 years back, and because of which it was really a pleasure to welcome Dr. Nitin Chitranshi. Right now he's in Sydney. So I will introduce you about Dr. Chitranshi and his latest work, what he's been up to. And before that, I can see that some are new participants. So as we always first talk about the company, what we do, how we established, and what are the programs that we are running, and why we have launched this program, Envision with BDG, under which you are getting these free sessions by renowned scientists and professionals from all over the world. So this is one thing which I would like to mention. However, people are still coming and joining the Zoom meeting. So let me begin with the introduction of the company, and then I will proceed with the introduction of the speaker, and then I will hand over to the speaker, and then we can continue the session. So BDG Life Sciences, that is Biodiscovery Group Life Sciences OPC Private Limited. We started this company 10 years back in the form of group, and that is why still there is BDG, that is Biodiscovery Group. We have not sorry, group we have not removed from the name of the company, because this is who we are. This is who we were when we started this company. Group, it is for all, it is for everyone, it is for the enlightenment, sharing knowledge and helping everyone. So this is the aim of BDG, that is Biodiscovery Group. This was the aim when we started this organization in 2009, and till today it is the same aim of the organization, to support you, to help you, to help in your career, to help solve your queries, and try to show you the right path and provide you quality trainings. Now everyone is providing training these days, and lately, since this COVID-19 pandemic started, everyone is now offering bioinformatics trainings. But then you need to be very much careful to which place you are going for your trainings, to which you are, because you will be investing your money, definitely, but then most importantly you will be investing your time. So rather than going ahead with the a training or a workshop or any programs, you should choose wisely. So I will not guide you on that. You are wise enough, and you are grown ups. Mostly we have students over here in this group, and we have even research scholars from different parts of India as well as from around the world. So you guys are wise enough to decide that where you want to have your training, and which at which place you will be getting your quality work. So continuing with my presentation, you could say that every day I discuss about being a guide, because this is who we are, because this is who we are and how we started this organization. So we are who will help you in solving your academic problems. How? Showing you the right path, solving your queries, giving you correct training, paving your path for a successful career, and giving you launch pad to excel into the field of science. So these are some very important points which which is the basis of biodiscovery group. So what was the need of starting the company back in 2009? The need of starting the company, and that is, a group, because we started as a group with a motive to help people, especially the students who were bachelors and masters. And why? Because they, at certain point of their bachelor degree or their master degree, they come to a point that they do not know what is the next step, especially in our field, that is the biotechnology or bioinformatics field. People are really not much sure that what they will be doing next, so they can have much better job opportunities, or how they can get a good PhD position. So this was the motive of starting the organization, to help, to guide, and basically to consult them that what is right for you. So, and obviously another thing was that there was talent, but then, and even the opportunities were there back in 2009. But then by the time the opportunity reaches the talent, the opportunity is gone, or vice versa. By the time the talent reaches the opportunity, the opportunity is gone. The reason was that there was a huge gap between opportunity and the talent. So we tried to fill that gap by creating the group. This group was created for students, researchers, scientists, and professionals who were related to the field of biotechnology, bioinformatics, and pharma. And what this group served, this group used to have free consultation, tutorials, books, assignments, and most importantly, job offers in India and abroad, PhD scholarships, fully funded PhDs, updates, research news, and latest science and technology news, and much more. Now, why I am saying was, why I am using past tense, because previously when we started this group was on Google and Yahoo groups. But then, there was no Yahoo group was no longer there, and then even Google groups is also no longer there. So then we converted this to our Facebook and LinkedIn and Twitter. So a dedicated team of BDG Lifesciences works on these platforms where you can post your queries, where you can ask ask for help, and people are there to solve your queries. So this is how we are helping now. And then we have, as of now, in my next slide, I would like to show you that as of now, we have 1 million subscribers from all over the world. So these subscribers are definitely from the Google groups, the Yahoo group, as well as Facebook. And then definitely we have a join us page on our website where where you can go and provide your information, and you can subscribe for the newsletter. So we will keep on sending you newsletters, and the same email address you can definitely write back and post your query. So these are some of our subscriber platforms. You can see LinkedIn is there, for example, 10%, mostly Facebook, because we cater a lot of students. So the students these days, they are on Facebook more. Then YouTube subscribers, we have Twitter followers and Instagram followers. Then on Messenger, we have WhatsApp, Google Duo, Telegram, and Botayam. And then on others, you could see that Pinterest and Flickrs. And then website subscriber, that is 70%, because as I have already told you, that we have a join us page on our website where you can just provide the information and you can subscribe to our newsletter. Now coming back to the company, because this is another part. One part is we are helping. Another part is we are doing business. What business we are doing? That I would like to tell you now, that we are a bioinformatics company, and our major clientele is bioinformatics and biotechnology and pharmaceutical companies, as well as clients. And what we do in this, we do contract research. You tell us about that you want to do certain, for example, say carry out molecular simulations and dynamics for your protein ligand complexes, because these days we are doing a lot of work on COVID-19. and we are working on sorry, people are sending our protein ligand complexes of M-Pro from SARS-CoV-2 and 3CL-Pro from SARS-CoV-2 combined to a ligand, and then we are doing the simulations, dynamics, as well as molecular docking studies. Then conducting workshops, e-workshops, that is online workshop, research projects, internships, training, webinars, seminar, and conferences. Now, I would like to point out one thing over here that, sorry, we are not new to this online mode of conducting the trainings and the programs. Basically, why? The reason is that we are doing it since 2012, and we very well know that how the online mode of teaching or online mode of research, because back then when we used to say that research project online, and then people were used to say how research is possible online, but then we have made it possible. We are doing it since then, and then, as you could see, I'm talking about this program, the research projects program. People from all over the world have participated, and as of now we have completed more than 60 research projects, and all of them have got international publications. So the research project program is especially for you to strengthen your profile, because these are novel research projects, and then on successful completion of the research project, it will be published at international level in the form of an abstract or oral presentation at conference or paper presentation at conference, and then later on into the journal. So basically this program is a win-win situation for both of us, the company as well as you, because we are launching the project and we are doing business. However, you are working on the project, learning the technology, and definitely you are getting an international publication, which is ultimately strengthening your profile, so you can have better opportunities for jobs as well as PhD opportunities for your future. This is what the bars of our business as of now. Research projects, it's at the top, because this is a program which everyone wants to strengthen their profile, so to have much better opportunities. So this is the best program of BDG Life Sciences. Then workshops, on-site workshops, like before the COVID-19 pandemic started, we have conducted more than 500 plus workshops around the world. Now, for example, if you name the countries like Mexico, we are there. In Africa, Sudan, Egypt, Nigeria, Kenya. Then you come to Gulf. Over there, we have conducted workshops in Dubai. Then you come to Thailand. There we have conducted workshops in Bangkok. Then you come to Malaysia. There we have conducted workshops in Kuala Lumpur. So we are doing a lot of we were doing a lot of workshops online around the world, on-site around the world. However, since the COVID-19 pandemic started, we definitely transformed ourselves, because we had workshops in Amsterdam, we had workshop in Prague, we had workshop in Singapore, and all these three workshops we have postponed it because of the COVID-19 pandemic. But hopefully, I hope and I pray that this pandemic goes away fast, so that we can come back to our normal life and we can have a good learning experience. So then we started this online workshop since 2012, as I was telling you. So in this, again, as it was online, so people from all over the parts of the world have been participating in our workshop. That is why you could see that this bar is, a little less. But then this is the second best program of BDG Life Sciences, the courses. Now, coming to the courses, these courses are free courses, totally free courses, which we have been doing previously also. But then before the COVID-19 pandemic, we used to do, say, for example, 1 or 2 free course in 3 months. But then since February 2020, as a gift for the 10th anniversary, because this year also is the 10th anniversary of BDG Life Sciences. We've completed 10 years in March 2020. So just to celebrate the 10th anniversary of BDG Life Sciences, we've started doing free courses. You'll be amazed to know that as of now, we've conducted more than 70 free courses on different topics: transcriptomics, cytogenetics, cytomics, nanobiotechnology, proteogenomics, NGS in food technology, next generation sequencing. There are number immunoinformatics, vaccine design, reverse vaccinology. There are number pharmacogenomics. There are number of courses which we have done. But as in total, I would like to say that we have completed or conducted more than 70 courses in which more than 14,000 participants from all over the world have participated. So this is what we have given to to the society, because it is all free, and it was still free. You can just check out the courses page, and there are some courses which are there, so you can go ahead and you can register for it. Then we have others is like contract research and project consultancy and project designing. So after doing 6 months, or I should say 7 months of free courses, innovation and creativity is an integral part of BDG Life Sciences. Hence, we thought that why not to do something extra? And that is the reason why you are here right now. Because that is the point where we decided that, okay, fine, we will gather renowned professionals from all over the world and ask them to speak on their topic of interest. So that is how Envision with BDG initiative has was born, or I should say it came into existence, because we wanted to give you more. We wanted to give students or research scholars more, so that you can avail in these hard times. You can avail as much as you can. So that is how Envision with BDG Life Sciences initiative came into existence. So our mission is to provide solutions for the future, definitely, because that is what our tagline is: solutions for the healthy future. How? By catalyzing the process of biomedical sciences into therapeutic technologies that benefit human health and society, and eventually fuel the economic growth. So basically, we are working towards the benefit of the human health as well as the society. And definitely this is the point which will definitely help in the economic development. I'll just quickly go through the services, or I should say the topics in which we are offering our trainings, as well as research projects and workshops. So you could see bioinformatics, basic and advanced. Right now, there is a workshop which is going on bioinformatics for beginners, and another workshop is also going on, which is on molecular docking. Then we have gene and genome editing using CRISPR, Cas9, Cas12a, Cas13a nucleases, and then you could see drug discovery, MD simulations, ADMET. Then in NGS, we are doing workshops and training in almost all kinds of next generation sequencing data analysis. However, we have not included scRNA and lnRNA, and as well as methyl-seq in this. But then we have started immune RepSeq, which is something very new. That is immune repertoire sequencing. So this is a unique kind of training program. Then yes, this is the program which has got a huge response: artificial intelligence and machine learning and biology. So we are doing a lot of programs in artificial intelligence and drug discovery. Then we have cancer, HIV, diabetes, and tuberculosis research. Basically, all I can say over here is that we are conducting targeted workshops having cancer, HIV, diabetes, and tuberculosis in mind, like cancer molecular targets, HIV targets, diabetes targets, where we are working on PPR gamma and S1P1. So there are a number of drug targets on which we are working, focusing on these 4 diseases. Value proposition is something very important, because I would like to point out, as in my presentation for the last session also, I want like to I've pointed out that innovation, then ideas, then service, and value. So these are some very important and strong words. Why? Because innovation is to translate ideas into service that creates value. So we are launching new projects, new programs, combining it with the new method of teaching, means the ideas, and then giving it to you so that it can have value in your life, in your career. How? Practical application of technology. We learn by doing the things. Everyone knows that we learn by doing the things. And that is the reason since 2012, we started our first workshop. We have made sure that whatever number of participants will come into our workshop, whether it is one or whether it is 150 or whether it is 200, everyone should perform on their own computer system, because that is how we will make you learn. Then hands-on training sessions. Every program, every training which we have has hands-on training sessions. Then novel research projects. Novel research projects on topics on which nobody else has ever worked before, because that is the reason how you will get international publication. And then result driven research, and then everlasting relationship. Everlasting relationship is very important, or I should say relationship is very important, because we will bond with you. We will people who have joined us in 2009, they are still connected with us, and we are still helping them, and now they have reached to a point in their career where they are helping. How they are helping? For example, in 2009, they were when they joined biodiscovery group of BDG Lifesciences, they were master's student or they were doing PhD. But right now they have become, for example, dean of a university or head of the department or a big scientist working in a company. So how they will help? They will help in form of employment for those who have recently joined us. So that is how it works. That is how, you know, you will also work. For example, you joined with us now, you are a master's student, and after 10 years you will become a big scientist. So at that particular time you will say, I need someone for employment, or I need someone for, I have a PhD position, do you have anyone? Why? Because in 10 years you would see that what is the, in fact not even 10 years, the first program you will do with us, you will see the quality of our work. And that is the basic criteria, that we provide quality work, so that you understand that how are the programs which are being offered at BDG Life Sciences and what is the level of the program. So you know that people who learn from here, they will have a very different kind of approach towards learning and understanding and level of knowledge. So we recommend those people, and we are providing jobs. So this is how we are providing job recommendations to the people. Now, not in our advertisements, we are highlighting a job assurance program, or if you do this training, or if you do this training program or workshop or research project, we will give you 100% job assistance. 100% job assistance, I don't think so that it is it is a true thing. But then maybe it might be, but then it doesn't work every time. So but that is the reason in any of our marketing posters or brochures or leaflets, you would not see that 100% job assistance has been given. So that is because we are not doing it. We are working on strengthening your profile, teaching you new skills, so that you can have much better opportunities in future, because this is what we are doing, and this is what we are promising, and this is what we are marketing. So that is why you should come to BDG Life Sciences. Next point you could see over here. I will quickly pass through this slide, because this slide tells that this graph is going up. It's from 2010, because officially we started our services in 2010. So this graph is growing up. Now, if we would not have been doing anything innovative, or we were not being creative, then at one point of time, this graph would have come to stagnant, and then, straight line, and then will come down. Because you cannot do business with the same idea. You have to keep on innovating. And that is the reason you could see that this graph is going up, which is a proof that we have been doing something innovative and creative in these years. So just to summarize, we have 10 plus years of experience in trainings and workshops from around the world, 60 plus research projects which we have completed, on which students, company professionals, and scientists have worked. Scientists, why I'm saying scientists, because someone who want to add more weightage to their CV by working on drug discovery research project, and add variety to their profile, that is why the scientists are working on our research projects. And as it is online, so anyone can come. And then most importantly, major, these projects can also be done as major research projects or dissertation projects, because we are offering these days 2 months and 3 months, and a lot of students from from India itself, they are working because these days they have to do online research, so that is why they are working with on our research projects. Then 400 plus workshops in India, 20 plus workshops in other countries. I've already told you the name of the countries where we have conducted. And then you could see that 50 plus workshops regularly we are doing at All India Institute of Medical Sciences, New Delhi, then IIT Guwahati, NIT, VIT, MMC, JIPMER, etc. The last point is happy customers, because this is ultimately our goal. This is ultimately what we want. Now, previously, when we started the organization, it was sorry, written feedback. We used to give the feedback forms. Then it was that people used to post it directly on our website. But now, since the last 2 years, what we have done, we have kept an innovative way of posting the feedback. Everyone can now, whoever the participant is, they make their video and we post it on our YouTube channel, and then we post it on our Instagram. So video feedback is something very important, because this tells you that what the participants feel in real time about the program. So this is valid for all the programs, especially the technical programs, basically the programs for which you pay. So that is why happy customers is the most important point. And that makes this organization, or I should say the step of communication, is completed when we get the feedback. So that is one thing. Now just would like to point out that we were the first one to introduce online training and drug discovery in designing. We are also the first one to conduct online and on-site training in gene and genome editing using CRISPR-Cas9. Now, Jennifer Doudna and Dr. Charpentier has won the Nobel Prize for this particular technology, for inventing this particular technology. However, we were the first one to conduct the training on these programs, which were in silico definitely, like for example, designing the PAM sequences, the off-target and the on-target analysis. So my particular expertise is drug discovery and gene and genome editing using CRISPR-Cas9 and NGS. So I am myself thinking that why not I should be taking the session, but then that I will see after 31st of October, because till 31st of October, in fact, I have contacted only 10% people in my connections, and mostly all of them have replied, and that is the reason why you are here, and every day, and then today also. So I will see that if I can take the session of maybe 1 hour or 90 minutes on these three technologies of my expertise, that is drug discovery technology, gene and genome editing using CRISPR-Cas9, and next generation sequencing data analysis. So let's see how we can go ahead. So keep an eye on our website. Then free courses, I have already told you that we've conducted more than 70 free courses during this COVID-19 pandemic, which we started before the lockdown. And as of now, we've conducted more than 70 free courses, and in which more than 14,000 participants from all over the world have participated. Services we are offering in virtual screening, molecular docking, MD simulations, predictive toxicology, and ADMIT, NGS data analysis, gene and genome editing using CRISPR-Cas9. Personalized training, we are offering personalized training for those who want to learn specific topics. So you tell us the topics and we will design the training program according to your need. Then research project consultancy. Research project consultancy is for those who who are in their PhDs and who stuck somewhere and they do not know how to proceed. So they tell us and we decide the future course of their particular program. So that is how we are doing and running BDG Lifesciences since 10 years. And you can take a screenshot of this particular slide where you could follow us. And I will ask my team to keep sending the links in the chat box. Now, one more thing, the feedback link of this particular session, which will be done now, will be posted into the chat box of Zoom only. Now, if you leave in the middle, then you won't be able to join it. So, and if you will not be able to provide the feedback, then unfortunately you will not get the certificate. So please make sure that you remain here until the end, for the full session. And at the end, you will, we will post the feedback link, and then you fill the feedback, give us your valuable comments, and then your certificate will be posted tomorrow. So this is one thing. And if you want this session, then you can subscribe our YouTube channel, because this session is also live on our YouTube channel. So you can go ahead and once you subscribe it, this session will be available over there, so you can view it later also. So this is about BDG Life Sciences. Now I would like to tell you about the today's session and about the speaker. I'll introduce the speaker, and then I'll hand over to Dr. Nitin Chitranshi. So sorry. So today's session is on brain investigation through eye, and basically understanding the molecular mechanisms in neurodegenerative disorders. So this session is conducted, as I have already told you, by my mentor and one of the very important person into my professional life. So I'm thankful to the, for Nitin Chitranshi to be here. When he said, when I asked him that you need to take a session, he, on at first instance only, he said that okay, fine, no problem, definitely I'll do it. So Dr. Nitin Chitranshi is a PhD and is a lecturer at Faculty of Medicine, Health and Human Sciences, Macquarie University, New South Wales, Sydney, Australia. He has a PhD in clinical medicine, and over the past 6 years has established himself as a leading vision neuroscience researcher and teacher. He has worked and gained experience in several areas of research, including pharmacology, pharmacognosy, and pharmacoinformatics. Dr. Chitranshi has also been the research intern at the Swiss Institute of Bioinformatics in Switzerland. He has more than 35 research publications and presented in more than 50 national and international conferences. Basically, he's a man who has taught me drug discovery and now is teaching about neuroscience. So I would like to welcome Dr. Nitin Chitranshi, and I would like to hand over this session to Dr. Nitin Chitranshi, and you can continue from here.
Thank you very much, Asif. It was a nice meeting you after being a long time. I remember the day when you and some of your friends came to do a project in a bioinformatics institute, and you have a project. And I remember the way you guys have take up the project and learn the things so quickly. And all you remember your friends, they are at a very good positions at the moment. It's been like more than 12 years, and I'm very happy to see you to leading this company. And like I'm just a small mentor, and it's been my small part what I played in your life to be a company CEO or MD for biodiscovery group. It's all your efforts and all your yeah, you can say your struggle that you did to reach this company and to bring it at this position now. And thank you very much for introducing me. Yeah, my name is Nitin Chitranshi, and my background is pharmacy. I did bachelor's and a master's in pharmacy from India. But as a major in pharmacy, I did pharmacoinformatics. So as Asif mentioned, I taught him about drug discovery things. So that was my major project that I did it in Central Drug Research Institute somewhere in 2007 and 8. So it's been a long time. And I was in India, I was into teaching, I was teaching pharmacy students. But in 2014, I moved to Sydney in Macquarie University to join a full-time PhD program in clinical medicine. So it's a big transition from like computer to the lab. Whatever we are doing on a computers, that's what we currently I'm doing it in the lab, like working on animals and neurons and a target and identifying the drug molecules, seeing the docking, what you do in bioinformatics, but we see how the drug or agonists bind to a receptor and how it activates and what are the downstream signaling. Those all I'm seeing it in the real life using animal as a model. So yeah, bioinformatics is a very important tool, and is a very important field for a drug discovery process to have like a complement process, or you can say it enhances the speed of a drug discovery, where you are not wasting your time in the lab, but you have some potential ideas. Okay, if we go for these particular set of molecules, we can find some good hits against this particular target. So like various approaches you can use: virtual screening, pharmacophore design, 3D QSAR, and docking simulations. And finally, you can test those molecules either in vitro or in vivo, or maybe that can go to a clinical trials. So I'm sharing with your screen, starting with, just let me know if you all can see it. Is it visible?
Yes, sir.
Okay, alrighty. So I'll keep this somewhere down so that I can mm, minimize. Okay. Alrighty. So today's topic is brain investigation through eye, and it's, maybe you can say, a joint collaboration between Macquarie University and a biodiscovery group. So I'm a I've been a lecturer, recently been promoted to senior lecturer now. So so my position is senior lecturer at Department of Clinical Medicine in the Faculty of Medicine, Health and Human Sciences at Macquarie University, and we are associated with hospital. And my supervisor is an ophthalmologist, so he do research in glaucoma, and we do more into glaucoma and neurodegenerative disorders that is related to the brain. So all right, that was my introduction, and I'll start with the first slide, the background. Okay, so what I'm gonna, in this complete this lecture, I will be talking about retina. So as you know, we see the images and everything through the through eye, is all information's been translated through retina. So retina is a very important part of a body system, where you can visualize the real world, and you can create an image, and then you transfer the information. So like, not many people know about that retina is a part of a brain. During the time of a development, the retina arises as a ectodermal derivatives of the forebrain, and it have like very similar process of biochemical similarities that happens in the brain tissues. We all know that brain has a major part that involves the neurons. So these neurons are also present in retina. So retina has different neuronal cells that may be called as retinal ganglion cells, photoreceptor cells, bipolar cells, that we'll see in the next few slides. So like the other part of a body, retina also being composed of neurons, and they get the blood supplies directly from the from the veins and arteries, and you can visualize these blood supply through OCT system, which is called optical chromatography system, in which you can visualize the flow of blood inside the retina and the vessels. You can easily visualize. So the ophthalmologists used OCT angio basically to see the any blockage of blood into the retina is there or not, and it's a very like a important tool for the diagnosis of any sort of shortage of blood supply in the eye or in the retina. So with like the way with the science is going on, and we are into an advanced stage of technology, so we can use both humans and animal retinas directly, and we can image them as a in vivo and in retina. We can use as a model for the neurodegenerative disorders of the eye, that can be like a age related macular degeneration, which is called AMD, retinitis pigmentosa, glaucoma. This is my specialization, where we see the patients with glaucoma, those It's age-related disorders where patient start complaining about their vision become blurry and they are unable to see the complete, like they have issues in their daily activities, they cannot drive, and glaucoma is basically called a silent killer, and where the patient doesn't feel any pain, only the vision start going down like time to time. And also the neurodegenerative disorders that are associated with brain, such as Alzheimer's disease. So as you can see in this figure, it's a brain and eye, and onto the eye where the bracket and a black arrow are showing is the retinal structure. So retina is composed of, if we talk about like I just took laser. Okay. So if we talk about the layer of retina, then it starts with the inner layer, like an inner retina, which is called as nerve fiber layer. So nerve fiber layer contains the axons and goes to the optic nerve. Whereas the ganglion cell layer, the major processing of imaging things like passing of messages that happens to ganglion cell layer, then inner plexiform layer, inner nuclear layer, outer nuclear layer, outer plexiform layer, outer nuclear layer, and pigmentosa and RPE, which is the retinal pigment epithelial cells. So the major part of rods and cones are composed in the outer part of retina. So rods and cones are help in visualizing in dark and to identify the colors. But if in major part, like in neurodegenerative disorders, the retinal ganglion cells get affected and they start dying, and the death of retinal ganglion cells lead to the death of the axonal cells. So these axonal cells are the axons which goes to the brain, and at the back of the brain where the visual pathways is associated is get connected. But as you know that the we have like two eyes, right and left. So the eyes, both eyes, when they goes to the brain, it crossover, and that crossover is called an optic chiasma. In the optic chiasma area, where the right part of an image is formed in the left part of a brain, whereas the left part of eye image is formed in the right part of the brain. So it's like a vice versa. So whenever we have an infection in one eye, the doctor used to say, put an eye drop in both of the eye. That was the reason why when one eye get infected, there is most chances that the other eye will also get infected because of optic chiasm. So this can be explained by this figure easily. So this is like a human brain and an eye. So if we enlarge this image, then you can see how the two eyes been located in a different orbital and their optic nerves forming an optic chiasma and going to the other parts of the brain, that's the visual cortex. So let's give a little detail of for the eye, what are this part. So the area where the junctions occur, like where all the neurons unite together, it's called lamina cerebrosa. Then outer covering of a layer is the sclera eye. Then fovea, it's a blind spot where there is no blood supply. And a pupil, like people get dilated when we use like a tropicamide or any like drug which can increase the like iris can go up and increase a large part of eye to enter into the lens, and during that period of time it is avoided to dry because your vision becomes blurred. And next is a cornea. The outer covering is a cornea, and the lens is an important part again, which is required for adjusting and forming image at the back of the eye. And a choroid, this is the part which is very rich in blood vessels. So these choroid provide nutrient to the retina and to the eye. And then the important part or the neuronal part is the retina. And like these are the axons, and if you know the structure of a neuron, it have a dendrites and an axons. So over to the axons there are a sheet of myelin. So myelin sheet is important to protect the axons to get damaged from like activation of any infections, or maybe from autoimmune disorders, maybe from the macrophages. So over to the myelinated sheath, there are nodes of Renvoyer which are important for transfer of message quickly to one part to the other. And this is the optic chasm where I was talking about, like where the information from right and the left get interchanged. And this is the LGN region. This LGN region is the part on a brain system where the images are being processed. Like this is a very important, it's a visual pathway. So most of the neurodegenerative disorders where we see a delay in ganglion cell loss or the neuronal cell loss, we usually see these all things in the MRI. And these MRI can define, okay, this person is in which advanced stage. But in glaucoma, the LGN regions is not seen much, but there are other diseases like multiple sclerosis. It starts from the eye and goes up to the brain. So because it involves axonal damage and damage of the myelin sheet. Then these are the primary visual cortex regions where the back of the brain. So these regions where chemical messages take place, and many of the neurotrophins and the chemical messages that are formed being transported from optic chiasms to the eye. So this type of transport from brain to the eye is called the retrograde transport, whereas anything formed in the eye, because these are also the neuronal cells, and which goes to the brain, is called the anterograde. And this is optic nerve, and these are the uncrossed fibers which are before the optic chasm, and this is the optic tract, and these are the optic radiations. So most of the MRI scans, we see the loss of nerve fiber layer in these optic radiations area. So this is about how we study eye till brain, um, including that like changes in eye pressures, changes in visual fields, and changes in eye size, and any, if we are doing in animal studies, then what are the molecular changes? Then we correlate those things with the brain MRI scans. So so these are two eyes, and like right and left, and the image as I mentioned is being from the right eye image from the left side, and the left eye image from the right side. Okay, so this is a disease we are seeing at a glaucoma at the moment. If on the image you can see a boy carrying a football ball, but if a person is having a glaucoma, he cannot see his face. So usually this type of vision is called a tunnel vision, where you can see the periphery, but the central part of the vision is lost. So as you can see on this side, this is a normal optic nerve where all the axons from the ganglion cell layer being united together and goes to the optic nerve. And the region where these being formed is the optic disc. During the early glaucoma stage, you can see the formation of curve. This curve is called an optic disc curve. Sorry. So this optic curve shows that there is a decline in nerve fiber thickness, and when it's in advanced stage, this cup becomes larger, and this shows that there is a severe damage and the axonal number is very low. And when the ophthalmologists do the OCT scan, they can see these different layers, and when we do the measurement, then what we find that the retinal nerve fiber layer is gradually start uh, going like becoming thin, and that thinning of retinal nerve fiber layer is a risk factor for the stage for the glaucoma. So like this is an eye. When the doctor examines the eye, he can, from the eye, he can say something, okay, something is happening in the brain. And like OCT scan can be used to diagnose for AMD, which is age-related macular degeneration, where you might know that some of there is a formation of blood vessels that largely the blood vessels are formed due to the increase in vascular endothelial growth factors, and those blood vessels are like abruptly being distributed in the eye. And but we are lucky that we have anti-VEGF therapy. So if someone develop AMD, the like doctors and the physicians, they do the anti-VEGF therapy where the patient receive anti-VEGF injections every month. So there are few drugs like Eylea, Avastin, another few they have been used, and it has been there in India also. So it's a very great discovery. It's a monoclonal antibody which stop the VEGF formation. That antibody binds to the VEGF receptor, and it stop the proliferation of VEGF receptor, and there is no more new growth of blood vessels occur in the AMD conditions. There is another disease called retinitis pigmentosa. So retinitis pigmentosa is something related to the retinal pigment epithelial cells, which attach to the sclera, and this is start dying, and there is a formation of occlusion bodies. So that this for this retinitis pigmentosa, there is still we are in a process of gene therapy. More likely, very soon we have antiviral gene therapy for retinal pigmentosa. The results are very promising. In US, people are doing a lot on retinal pigmentosa, and they're using antiviral like AAV2, which is called human adeno-associated viral vectors, to modulate the genes in retinal pigment epithelial cells. Other disease like glaucoma and optic neuropathies. Optic neuropathies, that means like multiple sclerosis, neurite, what is called NMO, and neuromyelitis. So these are the different optic diseases that can be diagnosed through eye. Also, the current research is more focusing on the disease for the brain, like Alzheimer's, Parkinson's, and transsynaptic degenerative changes. So as we know that Alzheimer's, there is no such cure in Alzheimer's. And what happened in Alzheimer's, there is a formation of amyloid beta protein, which is a neurotoxic protein, and it reach out from the neurons and form a plaque. And these plaques are unsoluble, like non-soluble, and they cannot be removed out. When it like plaque is formed at a certain part of a brain, it leads to the damage to the other neurons, and people start losing their memory. So Alzheimer is basically the loss of memory, whereas Parkinson disease is related to the disease of movement disorders, sometimes we can say, because usually the tremor occurs, and the protein which is involved in Parkinson disease is a cyanonuclein. And this is a new concept at the moment, where we are using eye as a model to see the transsynaptic degenerative changes, which is related to the synaptic formation occurs between two neurons. You know that when we communicate a language. Hello. Okay. So when we communicate, a language is a medium between the two people. Same way, when the neurons they communicate, there is a formation of synapse through what is synapse? That is formed between the dendrite part of one neuron and the telodendrite part of the other neuron, where they secrete the chemical message, and that chemical message just be taken up by the post-trans synapse, and it's been transferred to one neuron to the other. But when there is a blockade at one position, then the other neuron start dying. That's what we are examining these things in a lab at the moment. All right. So next is like an animal model. What we can do, and what we can see. Like human, the animals are very similar in genomic structures. So the eye in the rats or in the mice, we examine through, like when we kill the rat, when we sacrifice the rat, we nucleate the eye and do the sectioning. So here you can see the staining, which is called hematooxylin and eosin stain, which is pink and black in color. So the lower part of here, this is a normal retina of a mice eye. You can see the black dot are the ganglion cell. The upper is a retinal nerve fiber layer. This is the inner plexiform layer. This is the inner nuclear layer. This is inner plexiform layer, outer nuclear layer, and the retina pigment epithelial cells. So this part is basically composed of photoreceptor cells, whereas the inner part of the retina is important for the disease like Alzheimer's or like glaucoma. But as you can see, the degenerative changes when the retina is not healthy, the thickness is reduced, the cells are dead, and these things we can observe in the disease conditions. On a lower side, you can see how the light is stressed. You know that we all use mobiles for a longer period of time, and the blue rays are like very like dangerous for the eye system. So in the lab, what we do, we expose the mice for a certain duration for high intensity. As you can see, the stress light is reducing the rhodopsin, and these rhodopsins are reduces with time, and these here you can see have reduced. These numbers are reduced and become shorter. So the even the light stress also can also causes the damage to the retina, and the photoreceptors when they're dead, they are not recovered. And RD6, these are the genetic model where we don't have to produce any light stress. These animal model gradually with the time they have reduction in rhodopsin in their retinal layer. But if we treat these cells with any neuroprotective agent like some drugs or with some nanoparticles or gene therapy, if that can preserve the like eye functions or you can say structures. So these models are very useful in our studies and are useful for the drug discoveries. So what are the other alternative approach if we don't have animal or if we don't have cells? So the alternative approach here is the bioinformatics, where BDG, the biodiscovery group, can be very useful. And like there's a protein and a protein being docked with a small molecule, and we can see whether this can inhibit or not. Other approaches are like system biology, where you can use cells, like you can isolate cells, and you can see like activation through different drug molecule, and where you can see in the wild type is something which is related to a control, and grab 14 knockout, ko stands here is a knockout where this grab 14 particular protein is absent. So at a time when the activation is given, these being activated at higher instance as compared to the wild type, because the wild type can constitute grab 14 protein, but this particular mice does not have the grab 14 protein, so its rate damage is very high. That can also be checked through the western blot. You can see the wild type, this protein has a higher content, and the knockout mice doesn't have in their retina. And when we use loading control to see whether we have loaded the protein sample on equal amount, so we do by using actin or GAP-DH to quantify our like a protein estimation to see how much the protein has been upregulated or downregulated in normal and in disease condition. Okay, so we have talked about glaucoma. So let's see what exactly glaucoma is. Glaucoma is defined as a neurodegenerative disease which is associated with increased intraocular pressure. You know, like if you have your parents or grandparents who experience little vision loss, usually when they go to the ophthalmologist, they will check with the thermometer, they'll check the eye pressure. So if the eye pressure is more than 22 mmHg or 24 mmHg, depending on the body weight and the size. So this pressure at the back of the eye, when the pressure developed, so it start pushing back the retinal ganglion cells, and these ganglion cells start dying and leads to the optic nerve head cupping. As you can see, difference between a healthy optic nerve and an eye with glaucoma. So there is a large cup size and less retinal ganglion cells and less axon cells. So these conditions are known as glaucoma, and it's a very like this disease. It happens with the age, and most of the our parents and grandparents, they experience these things in their life. So what the doctor recommend, they recommend some eye drops which lower down the pressure. And when the pressure is not lowered by the medicines, so the doctor do some small surgery where they put a cut onto the tabular meshwork, or they put some stent where the aqueous and a vitreous humor have shared some pressure, and the pressure is reduced. But this is not a permanent cure. Usually, gradually with the time, the pressures start developing. It may be due to genetic disorders. So they are at the moment few genes have been identified, like myocilin and optineurin, and others. They have play an important role in like exerting pressure to the eye. And these genes have like what we the researchers at different labs, we are trying to modulate these genes through the gene therapy or through the stem therapy, where we can have some cure for neurodegenerative disorders like glaucoma. So in animal model, how we induce glaucoma. So you can see, this is a rat eye, and it's got a needle that carries a microbead. These microbeads are like a fluoro gold microbeads, small, very small beads, and we inject these beads into the anterior part of the eye, which is called as like a aqueous humor. And when the beads are being injected, they get accumulated at the tabular meshwork, where the aqueous and a vitreous fluid is that maintains the pressure. So when these beads get attached to the like tabular meshwork, it start building the pressure, and when the pressure is built up, it start leading to the death of the ganglion cell. So this is the example of a wild type, and this example of a glaucoma model. As you can see, the ganglion cell layer is reduced. It's only the pressure only affects the inner part of the retina, not the other part, like the photoreceptor cells or any other cells. So this is a structural function. This model we have already established, and we have published many papers on this. And when we see the electrophysiology, as electrophysiology is related to the stimulus. When the stimulus is given to the eye, it responds. The brain response is recorded. We do this through like an electrophysiology machine, or you can instrument is Phoenix. We basically use from Phoenix lab. This is from US. So a small intense light is given to the animal retina, and the animal is under the unconscious state, because we give them anesthesia, and then we give the a small flash intensity. As you can see, the blue line is the control mice which have no microbead injections, which is where there is no glaucoma. But these mice, when they have been treated for 8 weeks, so every week the microbead injections are given. And when we at the end point, when the animal was sacrificed and we did the electrophysiology, we find there is a significant reduction in electrophysiology. So now we can see that we can have a model, and similar results we see in humans also. So the reason why we give 8 weeks for microbead injection is that sometimes our body is maintained in a way that it clears out any foreign particles. So during like tears and all, the microbeads are moved out from the eye, and the pressure starts reducing. But in 4 to 5 weeks, the pressure is maintained up to 24 to 26 mmHg in rats or in mice. And also we do the in vivo imaging. This is called the OCT scan, what I was talking about. You can see this central part is the optic nerve head, and this is in the control. This is basically from the human data, and the one which have a reduced optic nerve, or you can say the thinning of the optic nerve, is in a glaucoma condition. So clear differentiation can be seen in the disease condition. So this is a part which where I'm interested, and I'm doing this one. I did this part in my PhD. So what we did, I use a viral vector, which is called adeno-associated viral vector, and it carries a GFP, which is a green fluorescent protein. Our human body does not produce this protein, so we have encoded a green fluorescence protein using a specific promoter which can express a gene only in the retinal ganglion cells, not anywhere else. As you can see in the image, these are the small dots with the green color are the GFP expression, and when the retinal sections is carried out, you can see these are only labeled at the inner nuclear layer, or you can say the RGCs are only labeled. So these upper two images are the images of the flat mount. Flat mount is something where when the animal is sacrificed, we open the eye, take out the lens, vitreous, and there's a retina. And the retina is basically, if you'll see in a dark light, is red in color. But when we take out in the lab and it's got bleached, it's totally white, transparent, like you can say like a cloth. So so this type of okay, so this type of like things in experimental point of view will be very helpful in identifying the labeled cells. So if we give the gene therapy in the glaucoma conditions, we can also quantify how many ganglion cells have been lost. So if it is a glaucoma condition, you can see the decrease in the GFP protein. So this is how the we do some experiments in the lab, and this is a like a cutting edge technology we are using the moment here in Sydney, and we are the only one in basically, if we'll say in like New South Wales, but some part in the Perth of Western Australia, people are doing the gene therapy, but not targeting to glaucoma. But specifically, if we'll say we are the only one in working in the area of glaucoma by using a gene therapy. And for this, I've been awarded in 2017 as a young, like what was that award? A translational research award from the ARVO Society, which is a very big society for the vision research in like a word. So ARVO is a conference that happens every year in United States. But this time we missed because of COVID. Every year we go, we present our work, and I've been awarded for this gene therapy in Australia, in Brisbane. So like this is something we are doing very unique in comparison to the other people who are working in the area of neurodegenerative disorders. Okay, so what's next? All right, so this is example, and this is example of a pharmacological agent. Like bioinformatics totally based on a drug and a target. So here the drug is 7,8-DHF. DHF stands for dihydroflavonoids. This is basically presents in the most of the like fruits. If you take any of the fruits, so it is the natural flavonoid, and this natural flavonoid as a nutrient, if we take, it's helpful. So what we saw that there is a protein, there is a receptor which is called a TRAC-B. It stands tyrosine kinase B receptor present in retinal ganglion cell layer. So this receptor is present onto the surface of the retinal ganglion cell. Whenever a molecule is agonist, is binded to the receptor, it activate. So this studies I published in a bioinformatics also I've used 7,8-DHF and in other derivatives to see binding of the like a ligand binding domain of TRAC-B receptor, to see whether how it is bind and whether other dihydroflavonoids have a potential to bind TRAC-B receptor or to activate that receptor. So what we find when the 7,8-DHF is given in high intraocular pressure, like in a glaucoma model, we find it prevent, like there is a prevention of retinal ganglion cell function as well as structure as compared to the high intraocular pressure condition. So this is with the high intraocular pressure reduction, the amplitude of the STR, which is electrophysiology amplitudes, is reduced. But those animals with high intraocular pressure condition, when given with 7-ADHF, they have the protective effect. So even the pressure is there, but these are some small molecules can be useful to protect or to prolong the disease progression, and it can be used as a neuroprotective molecule in glaucoma. And this is what we have published in International Journal of Molecular Sciences, and we have shown through molecular modeling studies, and also we have validated these things in the lab, like animal model also. So that this is the mechanism what I have found out and I published in current neuropharmacology. So as you can see, the TrkB, that is a tyrosine kinase B receptor present in retinal ganglion cell, and its agonist like 7-ADHF, which is something we are giving it from outside, but also our body produce, which is called BDNF, is called brain-derived neurotropin factor. But when brain-derived neurotropin factor bind to TrkB receptor, it phosphorylate the internal domain. Like some you need some initiation. A friend, when we try to go for a party and you are like you say, okay, I don't want to go, but someone push you up and you initiate it, and then you become active and get ready for a party. Same way, when the BDNF binds to the TrkB, it gets phosphorylated and it accumulates other protein, and the downstream is possible and leads to the survival of the DNA. So finally, there are some signaling pathways like AKT, MAP kinase, RF. So I'm not going into details because these are itself is a big topic. But what we have identified that in glaucoma condition, the BDNF is present and the TrkB receptor gets saturated. It's unable to activate by itself. What was the reason behind it? We find that the SHIP2, that is it's a phosphatase. Whenever something in the body system, if you'll see in a hemostasis condition, anything which is activated get deactivated. So if you have a kinase, you will have a phosphatase. So they both remain in like a hemostasis condition. And what we find that this phosphatase become active and it stop pulling back, like it stop make you it just keep on dephosphorylate the TrkB receptor, and this dephosphorylation is stop the downstream signaling, whereas this deactivation of TrkB receptor causes an increase to the endoplasmic reticulum. So we know that within the cell there is endoplasmic reticulum where the ribosomes assembly are being attached together and they're involved in continuous production of proteins. So some of the proteins which are not formed properly, they remain, get unfolded and get accumulated inside the endoplasmic reticulum, which are known as unfolded protein response, that leads to the activation of different, you can say some other proteins which are attached in the endoplasmic reticulum. The important one is BIP. This like a binding interacting protein, it release out, and major other protein factors, the stress factors get increased, and these stress factors lead to the death of the cells, and that's what we say it's apoptosis effect of cell death, or like a necrosis, and cells are not able to recover. So what we did so far in this study, that we have tried to modulate this particular phosphatase, and then we have seen whether if modulating these, like inhibiting or knockdown, can prevent the glaucoma associated damage to the retinal ganglion cells or not. So these studies we have published in current neuropharmacology, molecular therapy. So still like a lot of publications on these mechanisms. So how we talk about brain and retinal ganglion cells. So if you can see, the brain have been associated with retina, and especially the retinal ganglion cells. So brain produce the neurotrophin factors. These neurotrophin factors are taken up by RGCs. So usually the nerve growth factors level is reduced, especially in glaucoma condition. The tyrosine kinase receptor activation is reduced. Caveolin, it's an adaptive protein which brings SHIP2 protein to act like a phosphatase. So these activity get increased. This is another like a suicidal receptor um, present on the RGC cells. So this receptor is signal is also increased. Endoplasmic reticulum stress is increased. Apoptotic pathway is increased. So that leads to the damage of the retinal ganglion cells in glaucoma conditions. And this is when the RGC is dead, it leads to the axonal degeneration, which affects the brain. Apart from neurotrophin factors, there are genetic changes also been involved. So, like neurotrophin factor 4, BDNF, caveolin 1 and 2, myocilin, optineurin, WDR36, and tubercular meshwork, colin 1. So, genetic factors are also being involved in glaucoma conditions. So, it's not only, this is as we see that the vision loss happen, but understanding the molecular mechanism is an important step where we can advise for a new drug discovery or new therapeutic approach or maybe a new therapeutic target where we can focus on and we can help the pharmaceutical industry to go for a drug development. Now let's see if we want to see the genetic approaches, so how we usually do. So the genetic disease is basically is an important part of our drug discovery, and we do not we do not what we say we do not overcome those, and we do not avoid those genetic things. So to do research in a genetic direction, what we do, we develop a knockout mice. So this is simple an example I'm showing here, but I'm not talking about which knockout mice these are. I think I've written some. Okay, these are the caveolin knockout mice. So these mice, if you see the electrophysiology response is much better in the wild type, that is in control conditions. But when the knockout mice on the same age time point, we saw there's a reduction, and also there's a reduction in the time point. You can see the normal amplitude is 40, whereas the knockout amplitude is reduced to just like a 50%, maybe less than a 50%. So what we can do, we can produce a knockout like a global knockout, maybe through simple having some simple cut on a protein. Maybe we can use a flox mice, or we can just use Cree to activate, or maybe it can be done through CRISPR technology. What Asif at the moment they are talking about in different projects, they are using the CRISPR technology to produce a genetic mice, and those mice can be our target specific. Suppose if we want to study the knockout mice only in the retina, we can specifically using CRISPR technology, we can knock out any protein in the retinal cells. Elsewhere, the protein is continuously can be produced. So depending on your experimental techniques or your experimental ideas or your experimental plan, what you want to what is your hypothesis and what you want to study. So as there are other mice, as I talk about, like BDNF, which is also is a very important protein, is a neurotrophin. It's regularly produced in the body, which is called brain-derived neurotrophin factor. So usually, if you do exercise, people have shown now that if we do more exercise, our BDNF level is high. Like the brain neurons start producing BDNF, and these BDNF, the neurons like it. So to make a neuron healthy, BDNF should be high in a condition. So like when you are doing more exercise, you feel like more fresh as compared to like sitting at home. So what we did here, we get a heterozygous mice. It is not completely knockout or not completely BDNF is not present. So if the BDNF is not present, it can lead to maybe damage, like complete damage to the neurons, and the animal cannot survive for a longer. So even the heterozygous mice, they show the slight reduction in the amplitude as compared to the wild type, and also the ganglion cell layer is reduced, but it's not significant. When these mice been kept for a longer period where the heterozygous BDNF is produced, then we observe that these mice have a reduction in the ganglion cell layer and also the reduction in amplitude. So if we compare, like when we kept the mice for a 5 month and then compare it for a 1 year, we find a significant loss. So even the BDNF is produced, but it's not genetically produced in a manner which is required, of which the TrkB receptor can be taken up and can involved in translational process. So these type of heterozygous production of BDNF can also lead to the damage to the eye function and structure. So as you can see, this is a genetic factor, and to elaborate these studies, what we did, we get the heterozygous mice which have only at a single nucleotide polymorphism. Like if you see the sequence of a BDNF which carries an amino acid, so we use a valine 66 being mutated with methionine. Then we have seen what happened to the genetic variant. Because why we have selected valine 66 methionine? Because in a human blood samples, we find some of the patients have heterozygous BDNF produced, and that is through the genome sequencing, we find at a position 66, it's get mutated, and this is how we have produced the like a mice single nucleotide polymorphism mice, and we have studied this for like a 3 years. One of my PhD student have worked on valine 66 methionine mutated BDNF mice, and she has shown a very promising result. Like even a single mutation can affect the retinal function in maybe in a structure, and also the changes in retinal ganglion cells. All right, so what we did later on, we have introduced, suppose a person who is having producing a BDNF, but that BDNF is having like a mutation at one point. If that patient is also having a glaucoma, so that conditions we have developed in mice. As you can see here, when the mice were given with the microbead injections, their amplitude is significantly reduced. This is a model only. We keep this model only for like 2 months, like 8 weeks, but when the BDNF heterozygous mice were given macrobid injections and they kept for you can say like two months, they also find a highest decrease in amplitude as compared to the wild type. So this shows that if the BDNF is not produced in the efficient way, or the BDNF is like heterozygously mutated, and the person have a glaucoma, the chances of retinal damage is much more higher in terms of retinal structure as well as retinal function. So what we have identified from these studies, we identified that BDNF is a critical molecule, or you can say it's a critical protein. I'll just reduce this one. Okay, and it plays an important role in preservation of. Okay, let me. Okay, it plays a. It plays a critical role in preservation of inner retina, both in glaucoma as well as normal condition. Unfortunately, as I mentioned in our experiments, that BDNF is present but is unable to activate TrkB. There are few therapies where the people have introduced BDNF through injections, and what they find, there was very little success has been made. Even the BDNF in glaucoma is given, the function and structural functions were very less. So we have come to an hypothesis, like if we do like a gene therapy, can this be like have a protective effect to the RGCs? So that's how my project originated. So what I did initially, like we have like eye, this is a animal eye. So we have injected, this area is called, this area, this is a lens and a cornea. At the back of the lens is a huge jelly-like mass, which is a vitreous. And at the back of the vitreous is attached a retina. So what we did, we have injected intravitreally a viral vector, and these viral vectors are taken up by retinal ganglion cells. Why retinal ganglion cells? Because these viral vectors have a specific promoter, and that promoter is called a CAG. And the CAG is a hybrid promoter of chicken beta actin and cytomegalovirus. So this promoter, when the virus, if it is traveled to any other part of the eye or to the systemic circulation, it unable to express because it has a specific neuronal promoter for retinal ganglion cells. So as you can see in the optic disc, the this region there, this is a like you can say the structure of retina, which have all different layers. The upper layer is a retinal ganglion cell, and these are the optic nerve fibers. So the virus get expressed to the retinal ganglion cells, not anywhere in the layer of like photoreceptor cells or in the inner nuclear layer. This I will show you my results that we have obtained. So this is how the fundus image look like when we inject viral vector. AAV stand here is a adeno-associated virus which carries a GFP. This is a GFP is a green fluorescent protein, and here in a control, this only have a PBS or saline injections without virus. So there is no GFP protein, but when the GFP injected, we can see the ganglion cells have taken up the GFP and they are expressing green color. The transgene here stands is a gene of interest that we want to modulate. Either we want to upregulate, then we put a sequence of gene, and if we want to downregulate, means we have to knock down, then we use shRNA. So the transgene here, what we are using at the moment are like a neuroserpin, PTPN11, amyloid precursor protein, tau protein, if you want to upregulate and downregulate in the eye. So these are our results that we have obtained in a lab from the wild type and the knockout mice. So this shows that gene therapy has a potential to make changes, and we can make changes of a gene. We can modulate gene in the retinal ganglion cells in the disease condition through viral vectors. So this is an animal OCT scan where when we injected the viral vector, so the viral vector is not causing any damage, or you can see it does not making any differences in the different layers when we compare it with the control, like inner plexiform layer, inner nuclear layer, outer nuclear layer, or whole retina. So there is no significant differences in the layer thickness at any time point when the virus is injected as compared to the non-viral injected retinas. Okay, so then in my PhD, I've used this SHIP2, which is called, also known as being coded by PTPN11, and it's a phosphatase that we discussed previously. So what I did, I have knocked down this protein and also overexpressed this protein in normal condition and in glaucoma conditions. So while knocking down this protein in glaucoma condition, we find a neuroprotection. So as just a brief recap, what we have seen so far, that neurotrophin, when it binds to the receptor present onto the surface of the eye called the tyrosine kinase B, it activates activate the tyrosine receptor and undergoes a downstream signaling pathway, which is important for cell survival. But because of Okay, but because of a phosphatase and a feedback mechanism, this phosphatase is not allowing, its activity is not allowing the activation of TrkB receptor in glaucoma condition. So what we have seen, we have optic nerve exotomy is something where what we do, we just open up the animal eye at the back of the eye, we expose the optic nerve, and either we cut it down by using a scissor, or maybe we pinch the optic nerve so that the fibers get dead. So during that condition also the SHIP2 activity is increased, and also we have find when it's in glaucoma, the SHIP2 activity is increased as compared to the normal. So then we try to do, if you know about like to see the protein-protein interaction, that you can easily see in silico studies, but in lab how it is done through, we do it through immunoprecipitation, where we precipitate using an antibody, we pull down particular protein, and with that protein the which are interacted get also pulled down. Just way in the same way when we say when we like we have to go for a party, or we say for a student, like I say to my friend, okay, we have planned to go for a party, and you say, okay, can we bring our friends also? They say, okay, let's bring other friends. So we come to know like other friends, friends of friends, then becomes friends. Same way, like in a protein, when they interact, they come out. When we pull one protein, with that different proteins come out, and these are the results of the immunoprecipitation, where the SHIP2 is added like an antibody, and you can see the TrkB is also being put on. This shows that these two proteins have an interaction in the normal condition, but the interaction is increased when compared in a disease condition like an optic nerve exotomy or increase in IOP. The vice versa is done with TrkB, where the TrkB is pulled down, and we find that SHIP2 is highly interacted in increased IOP condition rather than in optic nerve exotomy. So we did the gene therapy, and you can see here the retinal ganglion cell get expressed with GFP, and this is a layer like a control with only having a SHIP with only having a GFP. This having a GFP as well as a transgene, you can see the transgene SHIP2 is highly expressed taken at the retinal ganglion cell, and there is a knockdown, like we have completely knocked down the red protein here, that is a SHIP2 protein. So that's how we are modulating genes in the animal retina using a viral vector. We can overexpress and we can knock it down. So these are my results that I have obtained during my PhD work. So you can see this is a full retina. This is a rat retina. When this is exposed bigger, you can see the retinal ganglion cell layer having a GFP taken up. Anti-neuron is the neuronal cell. Does anyone has any issue? No. Okay. So anti-neuron are the neuronal cells, because the retinal ganglion cells are the neuronal cell. So we have used anti-neuron antibody, and then we have used GFP to see the ganglion cell that are taken up by the GFP, and also we did all staining like SHIP2 together, and these are some results where in overexpression you can see the yellow part is higher, that these only express in the ganglion cell layer, whereas knockdown it's completely absent. But in normal condition, the staining is okay. So what are our results? So what we have seen when I've like injected macrobeads in the normal rats, in the in the normal conditions, we find the STR is significantly goes down, and this leads to the damage to the ganglion cell. And but in the knockdown condition, when we have completely knocked down the SHIP2, there is no activity of SHIP2, we find the knockdown has a better you can say significant amplitude. So here the scramble, why we use the scramble viral vector, because the shRNA sequence is something is like a the RNA sequence which attach to the transcription site, and it is stop the translation, like no more protein will be formed. So why we use scramble? Because we want to say that this effect is only produced through the shRNA, not with the virus. So the shRNA sequence, we jumble it. Suppose the sequence have TAATTP. So if we change the sequence and reshuffle it, it's become nonsense sequence. It cannot interfere in any other like a translational process. So the scramble here is used as a control for the knockdown sequence. And also what we find, like it just act like a glaucoma, and there is also a significant reduction. But when the animal where this gene is completely knocked down, there is a higher amplitude of electrophysiology is seen in PSTR. And also these results correlate with the results uh, of a HNE staining, where you can see this is a control condition, this is a glaucoma condition, and this is again a glaucoma with viral vector called scramble, and this is a glaucoma with knockdown gene. So when they are compared, we find that the retinal ganglion cells was significantly produced, like significantly protected. So what we find that um, like the loss was reduced from 63 to 38% in glaucomatous eyes when compared to scramble and injected eye. And I'm happy to share that this study has been taken up by a pharmaceutical company, and they have started like a clinical trial for the gene therapy in glaucoma condition. So this is something which we are taking it like a translating our research into like a man for mankind. And if this is successful, then it will be like a game changer in glaucoma therapy. So this is a mechanism that we have proposed, that in glaucoma condition the phosphatase activity is higher, there is increase in endoplasmic reticulum stress, which lead to the loss of retinal ganglion cells. While using an AV viral vector, if we silence this gene in glaucoma condition, we can see there is activation of cellular signaling pathway, which is through TRAC-P, and can protect the retinal ganglion cells both from structure and a function. So this is something we have just talked about eye. So how we are going to talk about between eye and brain, so that we'll see in few other slides. So as this is a recently we have published this in Progressive Eye Disease Journal, which impact factor of 14. So here you can see how the different mechanisms that we have to look after, like if there is a retinal damage or a vision damage. So if you see there is a transsynaptic dysfunction, means like the presynapse, or maybe like a presynaptic neuron and the postsynaptic neuron, if they stop communicating, that can also lead to a damage. Another factor that I did in my PhD is about like neurotrophin dysregulation, where BDNF or other neurotrophin is unable to activate the TRAC-B receptor. It may be related to like a mitochondrial dysfunction. As you know that mitochondria is a powerhouse in the neuronal cell, and when it get overloaded with extra work, it forms of free radicals molecules, and these free radicals start killing the cell itself. Also in some diseases like pro-inflammatory changes, that is related to formation of TNF alpha, nucleotide factor, MSC2, where the microglial cell get activated and start killing the neuronal cells. So this is another example where how the neuronal cell can die if the nutrients is less, is not being transported from one neuron to another, or maybe added with a neurotoxic molecule like amyloid beta. It can lead to the damage to the neuronal cells. Also through the endoplasmic reticulum stress, where the autophagy mechanism is surrendered. So if you know about autophagy, it will be easy to correlate what I'm talking about. So autophagy is something when there is any sort of attack to the body system, or specific to the cell or more specific to the neurons, our cells wants to try to protect themselves. They start producing some chemicals, and those chemicals are being like autophagy molecules which start fighting. But when the autophagy is compromised, it leads to the induction of ROS, and the cells can undergo in apoptosis. Another recently identified these two proteins, which are tau and amyloid beta. So these two protein brings a link between eye and the brain, because in Alzheimer disease there is a insoluble deposits of amyloid beta occurs in the brain, and tau tangles are formed, which lead to the Alzheimer disease progression. And this is another slide where we have seen like a BDNF heterozygous mice with time. Black hair is for 1 year, the mice kept for the 1 year. The central part, the mice kept for 5 months, and they kept for 2 months. They have just been like they have not been treated with any microbeads, or no experiment were done. With the time point, what we observe that the amyloid beta like 42 fragment is higher in retinal lysates of the BDNF heterozygous mice. So these give an indication that even a dysfunction of a neurotropin factors can lead to increase in AD-like pathology in the retina. So we find that the amyloid beta concentration of 42 fragment is higher in BDNF heterozygous mice where the BDNF is compromised, and it leads to the formation of maybe a plaque or maybe a neurotoxic, but still we don't know why this has been related. We're still investigating the correlation between eye and a brain through amyloid beta as a link in between. So what we find that increase in glaucoma also cause increase in amyloid beta protein in the eye. So in a wild type mice, when the intraocular pressure is increased, the amyloid beta concentration also increase as compared to the control. And BDNF heterozygous mice also have higher amyloid beta concentration in high IOP condition. Even this is a human sample. Human sample also, we have access to the human eye from our collaborator, the Lyme Eye Bank Institute in Sydney. So we have the control and the glaucoma eyes. So we took our retina lysates and just run for the antibody examination. What we find that in a glaucoma condition, the concentration of amyloid beta fragment is higher in human. This is our animal model in like a control and BDNF heterozygous mice. So overall, what we have concluded from these different sets of an experiment that increasing in intraocular pressure not only causing damage to the retinal ganglion cells, but also causing a neurotoxicity of amyloid beta fragments. So here comes like a two different brain I can show here is like a healthy brain and Alzheimer brain. It looks disgusting, but that's a true fact. So if you see the connection of brain with different regions is much more tighter, whereas in this in the Alzheimer disease brain, it's become like fissure, and there is a extensive loss of neurons. So what Alzheimer disease is? Alzheimer disease is a chronic progressive neurodegenerative disorders of the nervous system that results in severe cognitive loss of eventually death. So as you can see in a normal cell, these are the neuron in the brain. In Alzheimer disease, the brown patches are the amyloid beta plaques. So these plaques get deposited around the neurons, and it causes neurotoxicity. It prevents neurons for its functioning, like it cannot make a small connection between neuron to neuron. It may cause damage to the neuron by causing damage to the myelin sheath. It may cause damage to the axonal transport. So many mechanisms can happen in between in interactions with amyloid beta and a neuron. Here is an image of a slice section taken up from human brain with having a healthy brain slice and a severe Alzheimer. You can see the shrinkage and formation of fissure, So these how the small protein can itself cause damage to the brain in the Alzheimer condition. So this disease affects approximately 10% of individuals over the age of 65, and it is the fifth leading cause of death and major cause of disability in poor health worldwide. So what would be the reason why the Alzheimer disease are more prevalent as compared to the other infectious diseases like cancer or HIVs? The reason behind is that we have the expectancy because of the modern technology, advancement in science, and advances in drug discoveries. Uh, People have changed their lifestyles, and the lifespan, the death of Western people, if you see, they live longer than compared to the people in India. So they live like about like 90 or 90, 95. So these are the disease they died by because of disease of age related neurodegenerative changes. So it's become more prevalent now in western part of our country to identify the disease which is related to the age related disorders, because we have, for every disease we have some medicine that can be prevented, we got a surgeries. So people expectancy life expectancy is increased, and here in Australia the retirement age is 70. So up to 70 people are bit fine, they go for a walk, they are independent, they are not like the people those who are in disability or those who are dependent on others. And so if you see the Western countries like Australia, United States, Europe, they have more funding for Alzheimer disease as compared to the other diseases. So what happened in this disease ravages the brain and its cognitive ability to function. So when there are amyloid beta plaques are formed or tau tangles are formed, some cognitive dysfunctions get little changes. This is a good example of representative figures where you can see there is an axon, and this is a beta amyloid plaque, and this is another axon. You can see these green are the microglia or astrocytes that supports the neuron. So when these are found between the two neurons, the other neurons start dying if it doesn't get a nutrient. So these are the things that has been research have been trying to identify. If amyloid beta plaques are formed, then is there any medicine or is there any therapy which can clear it out from the brain, or there are any some inhibitors or any um, like like any compounds which prevent the sticking of amyloid beta fragments together. So so there are many different therapies are in research and going on, and people still try to find out how these plaques are formed in the brain. So by the time when the disease is diagnosed, it's become irreversible that this disease cannot be treated. Only we that worsen with the time. But there are some therapies, some cognitive changes that can be visualized. But with the use of like some medicine, like tacrine and donazepam that are being used, but it's not a preventive measures. So usually the diagnosis is done through the brain biopsy that happens after the death, and therefore the most clinician uses as a diagnosis of exclusion. So what happened in Alzheimer disease? There is a short-term memory loss. There is a loss of motor skills and language. Like people are unable to speak their words clearly. They even lost maybe a long term. They can remember. They can forget what's their spouse name. There's a disorientation, means they require some support, like how to stand up and to sit down. They are bedridden, and ultimately leads to the death. So critical that we develop the capacity to screen people and diagnose the disease before it manifests itself clinically in order to slow progression. So what we are doing here in us in our lab, we have our group. One of our group person are working on like human patients. So what we are visualizing the eye of the control and a disease patient, and using like a cognitive test. There are few questions they have been asked with the patients. There is an electrophysiology test. We measure the changes in retinal ganglion cell function. We ask the patient to give some bloods to see the genetic changes. So, and those patients are also undergoing 2 MRI scans. So these are like various aspects where we have collected a lot of data from the last 2.5 years, and we are correlating those results with the MRI brain changes and eye changes all together. So soon we'll come out with some good findings, and that will be a new expect to correlate eye and a brain together. So this is an image that I took it from a public domain. You can see how the neurons look like. So this is the cytoskeleton, which is the cell body, and this is the tail, or you can say the axons, and these are the telodendrites. So from the dendrites form a connection with the telodendrites, and the synapses are formed over there. So within the cytoskeleton, there is a nucleus membrane, mitochondria, endoplasmic reticulum, and everything, same as that are present in a cell. And over to the axons is the myelin sheet and a node of Renvire. So this myelin sheet prevents the axon to get damaged. But within the myelin sheet, like to support the axons, there are other cells present inside the system. What are those cells? Can be a macroglia, can be an astrocytes, or it can be other glial cells. So this supports the axons. But sometimes when the autoimmune disorder happens, like when the cells start fighting with each other, what they do, they start engulfing the myelin sheet, and these myelin sheet being engulfed by the macrophages, and the macroglial cells also, they start eating those. So when these myelin sheet are dead, it start progressing the disease for the axon, and the axon start dying. And this happen in anywhere in the body. It can happen in the brain, it can happen in the eye, or in the optic nerve. So the most common form of a disease with the myelin sheet where these happens is called the multiple sclerosis. So we have in collaboration with other professors who work in multiple sclerosis area, apart from Alzheimer's, glaucoma, and multiple sclerosis, as these all disease related to neuronal disorders. So let's see, this image is being presented by one of my PhD students. She put this in a thesis, and she has given me this image to show you today. As you can see, I was talking about visual pathway. So this is the eye and connected to the LGN region, which have optic radiation and the visual cortex. Happens in normal condition. So I mentioned about like retrograde and anterograde degeneration. So in the visual pathway, when there is a damage to the optic radiation, that the pathway get affected, and it leads to the damage to the vision or to the optic nerve. This is called retrograde, which originates from the brain and goes to the retina. Whereas anterograde, which is affected, like optic nerve get affected somewhere in between, maybe before optic chiasm or after optic chiasm, and it stop the progression or stop the transport of a material from the optic nerve to the brain. So this type of transsynaptic degeneration is called anterograde, and we are the one who have started this in the like in Australia itself, to see the transsynaptic degeneration both side, like retrograde and anterograde. So this is the retinal ganglion cells. You can see in healthy and dying condition, the red dots are the nutrients that are available in the healthy conditions. But if the target cells and these neurotrophins, they stop, they have not been transported, the cells die and obstructed. That leads to the dying of the RGCs. Now, we talk about, like we have seen many times, biomarkers, or what are the biomarkers. So we are identifying the ocular biomarkers to see changes in the brain. So what are those? It can be like eye movements, pupil flush responses, corneal nerve imaging, TF fluid, that the crystalline lens amyloid beta, and retinal nerve fiber layer thinning, which we have seen in glaucoma also, and optic nerve pallor, choroidal thinning, blood vessel changes. These are the different biomarkers that we are seeing at the moment in the retinal patients of Alzheimer's disease and comparing with the age match control. Also, like in a brain preclinical, we are interested in seeing the amyloid beta because that is a common link between eye and a brain, and how this protein is involved in synaptic functions and other different like axonal transports. So there are various approaches that we are using. We are using through topical biomarkers, through PET scan. PET scan is related to something like a MRI, and optic nerve examining through like OCTs, and functions using VP, which is called visual electrode potential. ERG is called electroretinogram, and some other movements. So these are the things that we are taking all together in account, and putting all the biomarkers that happens or that changes with the disease progression. Okay, so this is something interesting. You will see what this results is about. We have published these findings in 2016, and these are the initial findings when we identified there is something Alzheimer-like changes happens in the retina. So these are the transgenic mice. Transgenic, what I mean to say that these are the APP-PS1. These are the two protein which is get mutated. APP stands here for amyloid precursor protein, and PS1 stand for preselling proteins. So these are the proteins which are the hallmark in Alzheimer's disease condition. So we have like a transgenic mice that have overproductions of APP and PS1. So this is a flat mount that I was talking about. So as you can see, when we stain them with an antibody of amyloid beta or APP, we find small dots, whereas in the wild type, it's not present. And also, when we see in the amyloid beta protein, also we find some fluorescence. And in retinal images, the transgenic retina shows an increase in some like A beta or thioflavin staining is there as compared to the control one. So this is something which is immunofluorescence image confirm that there is amyloid beta present in APP-PS1 mice retina. So what we did, we other set of mice, we have taken out the full retina and run a Western blot. So as you can see, compared to the wild type, the transgenic mice has higher expression of amyloid beta as compared to the wild type or a control one. Also in brain, there is a higher expression of amyloid beta, APP-PS1, as compared to control. So this is a human data. This is a human eye, a control eye, and the human eye. So as you can see from the human eye, there is a higher increase in staining of amyloid beta as compared to the control one. These are some densitometric quantification being done between control and Alzheimer's disease patients. So we have seen that there is a deposition of amyloid beta. Can this amyloid beta deposition affect the structure and function? Yes, that's what we have seen. The APP-PS1 mice have a reduced electrophysiology, and there is a reduced like a thinning of retinal, complete retinal thinning, and also the loss of ganglion cell is much more higher in transgenic as compared to the wild type mice. So this is an interesting part where we are exactly not we are using, but other our collaborator in UCLA, that is University of California. They are using curcumin, which is a very important constituent of our Indian traditional that we use every day in our like food. So curcumin, they did, they have injected some curcumin in the patients. And this is a human data, right? This is a human data, what I'm showing here. So they've injected curcumin, and through systemic circulation, this curcumin get travel to the retina and get binded to the amyloid beta. So amyloid beta and curcumin complex. Here I've shown through molecular docking how this complex is formed. Since amyloid beta is a small peptide and curcumin itself is a small molecule. So here you can see that in human eyes and in animal eyes, both there are small dot can be visualized without taking off the eye sections or um, without any sacrificing or without any enucleation of the eye. So also the staining here, you can see the increase in curcumin and amyloid beta. E6E10 stands here for like amyloid beta antibody. So this antibody is higher, and this is, you can see the green is curcumin and the red is amyloid beta. So there is a complex between amyloid beta and curcumin in retina and also in extracellular deposits, because these are the mice, they are producing amyloid beta everywhere, not only in retina but everywhere. So again, these as I mentioned, there are two different proteins that plays a critical role in Alzheimer's disease progression. That is called tau. So the tau protein is a microtubule. This protein is like a microtubule protein, and it provides the stability. Suppose you have a few sticks, and those sticks, if you want to make it more tighter, what do? You tie it with a thread, and that becomes a strong type of like something which you cannot break it easily. So those threads are basically in microtubules are the tau protein. So those tau proteins in disease condition, they start losing out. So the stick is a microtubule and they are start separating. So the tubules get dead. So like what tau does, it's like a thread which is being tied over to these sticks, and those sticks now being opened up in a disease condition because of tau separates out. And this we have seen in Alzheimer's disease mice model that the phospho-tau is much more higher. So these mice have higher phospho-tau as compared to the phospho-tau in a normal condition. And also in human AD, we find the green is the phospho-tau that is higher in retinal ganglion cell, and this is a brain tissue. So like these are the results that confirming that there are some proteins which are involved in disease progression, and if we target those proteins can give an idea to go towards like a drug discovery, and it will be helpful for identification of a new targets. There's another technique that we all use is proteomics. As you can see, this is a fundus image. So what we do, we take out a full retina, either from the human tissues or maybe from control and disease. So we usually do like a OCTA, which is stands angiography, where we see the blood vessel changes. We do the hyperspectral imaging, and we can see correlate these things with the proteomics, and these can be used to see the changes with the brain data. So what we do in proteomics, like a mouse retina is taken, sample is prepared, protein mixtures. We run 1D electrophoresis, and the protein digested that from the small peptides. These samples are being inserted to the spectral machine, and the ions get separated out, and through the mass to ion ratio, the sequences have been separated, like can be identified, and through bioinformatics analysis we identify which of the proteins are being identified in this particular conditions, and we compare these with the control conditions. And like we can say, suppose okay, one of the protein is called amyloid beta, and in Alzheimer's disease we know it is upregulated. So we compare the amyloid beta peptides with the control amyloid beta, and we find it is upregulated. Some of the neurotrophins is downregulated. So proteomics gives a lot of information about different set of proteins that are playing an important role. But apart from that, it also gives an indications that which protein is upregulated and which protein is downregulated. So it's easy to identify. So that's how we go with the research. So this is a complete understanding that we have. This is what we have published recently. Eye and a brain correlation. As you can see, this part is an eye examination, and the other right-hand side is the brain examination. So for the eye examination, if any of the patients with complaining about like he has short vision and his memory is getting lost, then we do electrophysiology, we see the angiography, we do the visual field test to see the changes and contrast sensitivity. We also do the VEP, which is called visual electropotential. If you have seen any of your parents or grandparents having something electrode put onto the head, and a flash of light is given, and they might be have asked to look at the arrow and select if the arrow goes to the right, press right. If the arrow goes to the left, press left. So that has to do continuously. And there is also hyperspectral imaging, where the beam of light is passed to the eye and it is scanned. So this is what we do to the eye. When something on a brain correlation, same patients, we ask the patients to go for like a PET scan, where the imaging can be compared, and we can go for like fMRI, SMRI, or DTI. Then also a neuropsychological assessment. That assessment is related to like memory, speed, creativity, attention, flexibility, focusing. So we ask few questions, how the patient respond to that questions. We mark them, and then we correlate with these examination data. Also, there's EEG, FNRS, MEG, TM. So these are the complex techniques that are being used here to examine, and all these together we define our conclusion. So it is not only a one test that can confirm, okay, this person is having a neurological disorders. So we have to go through all different process. We have to integrate different approaches, and all together we do a diagnosis and a treatment. So this is something like a risk assessment, that is age, genetics, sex, lifestyle, and other comorbidities, amyloid beta plaques, and tau hyperphosphorylation. These are the two things that we take into account and leads to the AD diagnostics and a monitoring. All right, so I come to the conclusions. I think I've spoken a lot. So there is an overlap between glaucoma and Alzheimer's disease. Amyloid beta and tau protein, their deposition occur in the retina in glaucoma and Alzheimer's disease pathology. The retinal function recording, we do it through electrophysiology, imaging through OCD, and can provide a measurable and multidimensional indicators of the disease state, what state the disease is at the moment. And like retinal monitoring in conjugation with clinical examination can improve disease diagnosis and prognosis. And also the proteomic analysis will unravel spatial and temporal effects that are specific to the disease in the retina. So it's good to know what are different proteins and cellular pathways going up or down, that we can do it through proteomics. We can use like a retinal biomarkers which enable quantitative monitoring of the drug and lifestyle intervention effects in neurodegenerative disease. So there are references, and thank you all for the listening.
Thank you, Nitin sir. Thank you very much for your enlightening session, and it was really good that we have come to know about this cutting edge technology.
Mhm.
So now we will quickly, we had just had two questions, so I would just
Asif, it's been, can you just give me a minute? I'm getting a call from my wife. It's already been like 10 here.
Okay.
Yeah, I'll just be here in a moment.
Sure. Okay, participants. So the thing is that after a question and answer session, we will post the feedback link in the Zoom chat, and you can fill the feedback form. and the feedback form will be available or will be valid only till 11 p.m. tonight India time. So you have a lot of time to fill the feedback, and your certificate will be sent to you tomorrow. So the feedback link will be shared just now, and just after Dr. Nitin Chitranshi will answer two questions. We've got only two questions. So once he answers those questions, then we will put the feedback here.
Yeah.
Okay. So the first question is, sir, can you please explain what is knockout mice?
Okay. So knockout is something when we remove the gene. Suppose this example of a gene I can take about any gene that produce maybe take an example of an eye gene called rhodopsin, right? So rhodopsin is a protein and it's been encoded by a gene. So if the gene is completely gone, like there is no gene is present and no rhodopsin will be produced, so that is called a complete knockout. And we can make it like a conditional knockout, where we can make rhodopsin to be knockout in a specific cell, not anywhere else. So there are two different terms, like a global knockout, conditional knockout, or maybe like a knockout of a choice. So as Asif is showing more on CRISPR technology, so this CRISPR technology can be used a lot to generate different types of knockout mice. So did I answer the question?
Yes. And the next question is, thank you very much. So the next question is, can we use BDNF as liquid biomarker for any neurodegenerative disorders or glaucoma?
Yeah, we are using BDNF protein as a biomarker. How we are doing this at the moment? We are analyzing the blood sequences of like a blood analysis we are doing. So we are separating the blood cells and the plasma, and those plasma are being run through the proteomics studies, and any changes in BDNF is being noted in control and a disease condition. If that remain consistent, then definitely the BDNF is one of a target protein. We can say if this protein gets changes or get mutated, then the person particularly at certain period of time will have this particular disorder. So the biomarkers are something which is developed like we can use it to identify a disease progression in advance. So it is not something which is being you can say is like at a very large at a very last stage we can identify, and then we try to treat the disease. So biomarkers discovery are basically the discovery being used to have the identification at the earlier stages, rather than going for a disease progression and treating the disease, because there is no such disease which can be completely eliminated.
Okay.
Yes.
And then we've got two more questions, so I'll just quickly go through them. Gene therapy is the best method to diagnose and cure the disease glaucoma?
At the moment, yes. At the moment, and this is a very advanced, because we are using a viral vector to modulate gene is very specific to a specific region, not anywhere else.
Okay, good. And then the last question is that how to select a specific vector which will silence the target gene? There's a lot of vectors associated with several proteins.
Yeah, so selecting a viral vector is an important concept, because as you know that virus, inserting a virus itself, it's like an autoimmune, something where the our cells start producing some antibodies, and they start killing the viral vectors. But there are few viral vectors that are produced in a lab called human adeno-associated virus, which have a very like a sequence similar to that of human genome. And these are like they have till now it's not been seen any immunogenic response when this viral gene therapy has been used in the clinical trials. And also, I'll say that one of the inherited disease of the eye, where also the vision is lost, the viral gene therapy is been successful, and they have been continued in the different countries in US and Europe. The people are with confidence using a viral vectors to treat the disease. Even when we started working on a viral vectors, we got in touch with one of our professor working in a Westmead hospital. What he did, he generated like a liver cirrhosis in in a small child about like 7-8 years through viral vector. So he saved that kid's life, and he that person was the like a Australian of the year. So viral vectors, if it is like a friendly, and if it is not causing any damage to other cells, then these therapies are have a potential to go for a clinical trials and can modulate some cellular mechanisms that happens in the body in a disease condition, which we have to put a control on it. So yeah, viral vector, what I mentioned is a human adeno-associated virus, is a good virus. It's like a human friendly. It doesn't cause any immunogenic response. But there are other viruses like lentivirus. They can only use to study some part of a some mechanism, but they are not in the clinical trials at the moment.
Okay, so thank you, Dr. Nitin Chitranshi. It was a very nice session, and thank you very much for all the participants for their patience and for being here for this entire session. So thank you very much, sir, and thank you very much to all the participants. The feedback link has been posted into the chat box, so you can just click on it and provide your valuable feedback, and it would be valid only till 11 p.m. India time. So kindly, put your feedback. And I think this session we will conclude, and then definitely you can check out our workshops page for the upcoming session. So thank you very much, sir. Thank you very much to all the
Thank you,
participants.
Asif. All the best, man.
Thank you very much.
If you need any support, just let me know. I'll be there for you.
Thank you very much for your time and the nice presentation. Thank you.
Thank you very much. See you. Bye, participants. Bye.
Bye.